All right, so I'm going to talk about genomics. Please, again, tell me if I'm speaking too quickly or too loudly. Genomics analysis on Spark. And to start with, let me ask you a question. Have any of you ever actually done any bioinformatics? Are there bioinformaticians in the audience? I know there was a couple. Great. Have any of you ever taken a biology course ever? All right, so maybe a little bit broader. I'll be honest with you. I'm going to confess. The last bio course I took was high school biology. So everything I've learned is on the job. I did my graduate research in genomics. I've worked at Novartis and a couple other companies working on genomics infrastructure. I now work, among other places, I work at a company called Tamer, but I also work for the AMPLab, contributing to this project. But what that means is that before we get started, I'm going to spend one slide giving you a very brief introduction to genomics. I apologize. For those of you who know something about bioinformatics or genomics, this will be incredibly high level, and probably everything I say is wrong, but just bear with me. So as you may know, the genome is the blueprint for all the proteins in your cell. The proteins are the molecules that do stuff. Changes in your genome may be indicative of a propensity to get a disease or have a particular symptom that you may not like living with. So the thing that we do in genomics is that we want to understand your genome. And the way that that works is we go through a multi-step process. The first thing we do is we take a sample. Literally, we get pieces of your genome in a vial. And then we put that sample in a machine called a sequencer. And you can think of the sequencer as a black box. It takes in these samples of DNA in liquid, usually in liquid form. And it spits out basically a text file. Your genome, we think of it as a long string of A's, T's, G's, and C's. It's about 3 billion letters long. A, T, G, and C represent monomers, or pieces of molecule that we understand chemically. And the total sequence actually presumably will tell us something about you, your biology, diseases you may be suffering, other kinds of diseases like cancer, which are the last 20 years of cancer research have revealed that cancer is basically a disease of the genome. So understanding the genome of your tumor, if you have cancer, is an important step. So that's what the sequencer spits out, this line-by-line string. These are called reads of A's, T's, G's, and C's, these strings, hundreds of millions or billions of these things. And then bioinformatics or genomics is the process by which we basically reverse what we just did physically to gain some understanding about your genome. We reconstruct the data in those files. I won't go into how we do that. You can ask me about it offline if you really care. But the other thing I want to do is spend a moment talking a little bit about the conduct of genomics today. Also, can someone remember to give me like a 5-minute heads up when okay, yeah, so I really will move quickly. What does genomics look like today? And the thing I first want to tell you, and it's great that I came after the previous two talks because I think they also emphasized a lot of the structure of scientific computation in a lot of different places, in a lot of different fields. So genomics, like image science and other kinds of sciences, very often works with pipelines. This sort of example pipeline I've drawn up here on the right, multi-steps, branches and merges, many, many different stages of computation. And you have to go through all of this in a kind of batch mode in order to get the final result to understand something about the genome and maybe how it's related to a disease that a person or a group of people might be suffering from. The important thing to remember here is that each of these steps in these workflows are often they're written often by researchers today. So even if you go to a pharma company, you're often using tools that were written by a grad student somewhere. And these tools are written in all sorts of different languages, C, C++, Python, Java, Scala, Perl, R, MATLAB, the list goes on. It's almost infinite, as many languages as there are. And they're all written to basically use the same fundamental baseline of infrastructure that everybody has, which is the command line and the file system. So each one of these steps will typically output files that the next step uses as input. You go to and from the file system in custom file formats each time. So that's one component of the story in bioinformatics today. The other component is that the sequencers themselves, our ability to generate the data that goes into these pipelines, these sequencers are collected in a small number of locations, relatively speaking. So if you're in Cambridge, in Massachusetts, where I live, there's the Broad Institute, which is a big, they're sort of one of the larger collections of sequencers in the world. You've got various companies that have large collections of sequencers. There's a BGI in China. There's a small number of places that have the lion's share of sequencing capacity in the world today. And large computational analyses today talk about analyzing data in the size of terabytes to petabytes. So when you say we're going to analyze 5 petabytes of genomic data, that's a fairly large analysis today. Hand in hand with this fact that the data is often collected in batch, very often by researchers who are working on a publication or people at a pharma who are working on a distinct project. The computations are run in batch, and because all the intermediate results as well as the inputs and outputs are all files, data sharing is basically synonymous with data copying. If I want to share data with you, what we talk about is how I give you the files that I generated as part of my pipeline. And these workflow pipeline systems are everywhere. And that final point about data sharing being equivalent to data copying is one of the major pain points in bioinformatics today. This, and I'll read it from the back. I don't know if you can see it. This was a tweet I found about a year ago. That it was right after Amazon announced that they were going to experiment with with drones to do delivery for things. So this is someone from the Sanger Institute, a big research institution in the UK. He wrote, "Not wishing to be outdone by Amazon, Sanger Institute develops drone delivery systems for BAM files." BAM files are a canonical kind of data file in genomics. And it's a picture of a little mini quadcopter with a USB drive dangling underneath it. I found this intensely funny, but the sad part is that it's actually not really a joke. Someone mentioned shipping hard drives around. But bioinformaticians will ship each other collections of hard drives all the time. This is not a joke. This is the reality of data transfer. And subsequent to data transfer, the reality of the pipelines that we run on our data after we receive those hard disks. It's nuts. It's nuts for the following reason, because the thing that I didn't mention, this is a graph, by the way, that genomicists love to show. It's taken from the NIH. Basically, the sequencers, the engine that drives genomic analysis, they're undergoing a sort of Moore's law kind of evolution over the last 10 years on their own, maybe even super Moore's law. And then, depending on who you ask, maybe we've fallen off Moore's law already. So what this is showing over here is basically the cost of completely sequencing a human genome and how it's fallen over the years. And you've seen that white line is a Moore's law line. The green line is the drop over the last 10 years. This is driven by what genomicists will call next generation sequencing over the last 10 years. So the idea is that almost 15 years ago now when we completed the Human Genome Project, it was hundreds of millions or billions of dollars and 10 years to sequence something like a dozen individuals. Now if you run one of these big genome sequencing centers, if you amortize the cost and the time, you're basically sequencing a whole human genome in maybe a day for maybe $1,000 or $1,500. And that's going to continue going down. So I fully expect in 10 years to be able to walk into or maybe 15, to walk into a Walgreens and be able to sequence, maybe I won't, but theoretically, and I sequence a genome the same way that I used to get my photos developed, right? An hour sequencing or something like that. What it means is that sequencing is going to become decentralized. Everyone's going to have access to it. The data deluge is already, it's maybe not at our knees, maybe it's at our waist. It's not up here yet, but it's getting pretty close. There's not going to be any single place that has access to all the data at once. And shipping around hard drives is going to be completely infeasible. I mean, it might be infeasible already, but it's going to be completely infeasible. So what I'm interested in, and the reason I'm excited about Spark, is I'm interested in starting to think about what the next generation of genomic analysis architecture will look like. It's clear to me and to a number of other people in the field that simply writing single command line tools that read and write files from local disk on a single node at a time is not the kind of thing that's going to scale long term. Even today, it doesn't allow pharmas to do what they want to do, or large companies, which is basically turn the dial, throw more resources at it, and get more or faster results, and you can't do that. So how do we begin to solve that problem? So the first thing that I want to point out is that a lot of these pipelines, the successive stages are not independent. They share parallelism. So subsequent stages of the computation may all parallelize the reads in their data by sample. And at some point you may switch. So at some point you may start to parallelize your reads by a different axis, so maybe like genomic position. But that's basically a big sort. That's the kind of thing that we hope Spark will be able to do efficiently. Databricks likes to talk about how they break sorting records. That's really exciting to me as a person in genomics and bioinformatics. It also means that we can start thinking about a world in which we don't have to keep around every single intermediate file. We're not going to disk. We're not saving these files. We're not getting the sort of subsequent explosion in data size that people experience. And finally, I'll talk a little bit about this at the end. It allows us to separate concerns, methods developers on one hand and people who focus on performance and execution of the platform on the other. So the parallelizability story, the large-scale sorting, sharing of parallelization across stages, that's one part of the story. The other thing, and I'll personally put a plug in for this that I really like, is there's a whole constellation of interesting tools around Spark that are also incredibly useful for genomics. My favorite two are a pair that go really nicely hand in hand, Avro and Parquet. I know both of those have gotten shout outs in earlier talks. So Parquet is a really nice, efficient columnar data storage format. Avro is a language like Thrift or protocol buffers that allows us to express data models in a kind of language-independent way. I think of this for bioinformaticians much the way that no one writes machine code anymore. They write it in a higher level language that they understand, and then they compile it down to machine code or whatever system you're targeting. But bioinformaticians still love to define custom packed binary formats for their particular file data type of choice. I think that's also the kind of thing that really needs to go out the window in the long term. It's not scalable. It's hard to maintain. People are terrible at specifying file formats. And so Avro and Parquet, as an example, give us the ability to think about compiling our file formats from our data models, much the same way that we compile the code that actually runs on our machines from the code that we understand. So the main thing that I'm talking about today, I'm up here as a representative of a project called Atom. Atom is basically, we think of it as Spark for genomics. So much like the previous talk where we saw sort of a development of an imaging layer, a set of primitives that are callable from Spark that represent the kind of image analysis steps or stages or computations that you want to do. It's not all group by and flat map, but it's things that actually make sense to someone doing image analysis. That's what Atom is basically for genomics. So what we're trying to do is, in an open source, open way, re-implement some of the core bioinformatics analysis, genomics analysis routines, and core data manipulation primitives to run and to run efficiently on Spark. It's open source. It's Apache 2 licensed. It's hosted by UC Berkeley and the AMPLab. I'd be happy to talk about it with any of you guys after this talk. But the main thing that we're doing, an example of what you might find if you look inside of Atom, is a re-implementation of some of the core primitives that happen every day in different tools in genomics. So an example of this is what I'll call a spatial join or a join by overlap. You can think of it as a kind of 1D version of what you might find in a GIS system. So in this case, we think of the genome as basically defining a one-dimensional coordinate system. And different data sets manifest as point sets or ranges whose coordinates are defined along that coordinate system. So if you see the red and the pink boxes, I don't know if you can see them in the back, those might be two different data sets. Maybe the blue boxes, sorry, blue and pink. The blue boxes might be genes. The pink boxes might be regions of variation. And a join by overlap just simply does a join where you join pairs based on how they overlap in this one-dimensional coordinate system. The problem is that everybody uses this kind of primitive. And every tool you find, every different framework has a different implementation of it in a different language with different characteristics, with different assumptions about the data that goes into it and the way that it produces data on the outside. So what we're looking to do is really think about doing a distributed implementation of, for example, this kind of primitive and several others, and to do them efficiently on and in Spark. All right, so how am I doing on time? 5 minutes? All right, so I'm going to wrap up. And that's good, because I've only got a couple more slides. So the final thing I actually wanted to do was tell you, it's not just about like, oh, you know, hey, here's Spark, and oh, it's genomics, and it's got all these hard things. Let's just go rebuild genomics on Spark. It's never that easy. And I wanted to highlight this. This is, I'll read it to you if you can't read it in the back. This is another pair of tweets. And the top one is linking to an earlier version of this talk I gave at a Spark meetup. And he says, bioinformatics has a lot of workflow engines. And it's true, they have dozens. I think I did a count for one of my friends, and it was something like 18 or 20 that I could come up with that I knew of that were in use by people I knew, and I'm sure there's more. Bioinformatics has a lot of workflow engines. A new one is born every day. And the guy who responds to him, is a well-known bioinformatician himself, says, you know what we need? A meta workflow engine. Obviously, he's being sarcastic. So there's real pushback in the genomics community by people who basically are thinking, why are we writing a lot of workflow engines? Like, why are you spending your time building yet another, or using or implementing things on top of yet another workflow engine? Why can't we just focus on methods, answer the scientific questions that we need to answer today, and get going? So let me just point out, this is my only slide without an image, and then we're pretty much done. I think there's actually, there's good arguments on both sides, I'd argue. So in the no side of things, there's, we're talking about rewriting a lot of software. We're basically we would be taking up energy and oxygen from the people who are working on methods today. And a lot of people would say we don't understand the data as it is already. We should focus on understanding the data before we're talking about scaling the computation that occurs on top of it. From my perspective, I think it's a little bit different. I think there are good reasons to do this kind of thing. So the move to commodity computing or the cloud, I think that's an essential move. People are basically not going to be able to have locally or under their complete control the kinds of resources they need to do large-scale computation in the future on the size of data that's actually being generated by the machines they own. And again, like I said, I want to get to a world where sharing is not copying. And to do that, we actually need to start rethinking how some of the methods themselves are built and the platform on which they operate. But most importantly, I actually want to get to a world where I don't open up a bioinformatics tool, look at its source code, and find an implementation of a distributed or an out-of-core sort algorithm, or some other kind of low-level system programming, like managing the number of open file handles and building your own indices on top of your own custom-defined binary file formats. So these are all things that actually happen. I'd like to get to a world where people can work and focus on the platform, and people can focus on the method, and both of them can benefit from the work that the others are doing. And I think that Spark and a project like Atom is a great example of something that will allow us actually to get to that. It's a step in that direction. So the final thing I want to say, before, if we want to take a question or two, we can do that. But the final thing I want to say is, this is not just me standing up here and talking or waving my hand about work that we've done or that people have done. And I'll be honest, I've done a fraction of this work. I'm a contributor, but not by any means the only one or the biggest one. I want to say, I'm not just telling you about work we've done. I think if you probably did a show of hands, every one of us, or maybe most of us, have been touched by disease, or cancer, diseases that are heritable or that are diseases of the genome in our lives, or we know someone who has been. At the same time, each of us is carrying around in our pockets and on our laps computers that are millions of times more powerful than the computers that were developed to, the computers that were used to basically develop the hydrogen bomb and things like that. So for me, I think there's a very powerful incentive for those of us who work in high performance computing, who work on cluster computing, distributed computing platforms, the kinds of platforms that can support this kind of scientific analysis. I hope each of you think about the way that you might contribute a little bit to work like this in science, open work to support high throughput, high access science in fields like genomics that are really undergoing a data revolution. Atom is not just sort of a fixed in stone project, it's open source. And by that I mean we accept pull requests. So each and every one of you, if you want, today or tomorrow could go home and figure out some way that you could contribute and file a pull request, and we'll review it and we'll merge it. This stuff is done in the open, and it's best done with more of us. And this is the wave of the future in drug discovery, in basic biological research. If we do it right now, each of us will write code that helps diagnose a patient, discover a drug, or cure a disease. I think that's a really powerful message. So with that, thank you for hearing me talk. I hope I didn't talk too quickly, and I'd be happy to take a question or two.
Hi. Thank you. It was a wonderful talk. One of the problems.
Let's see.
Yeah, here I am.
Sorry, there you are.
Is that people in the genomics area have confidence in certain tools. If you come up with a new tool that's implemented better, how do you prove to them that's as good as the old tools?
Yeah. I mean, this is actually a problem that people in genomics have with their own tools. So the tools themselves will be updated or will not be updated anymore, will fall out of date. People have a very hard time even understanding what the ground truth is or how to compare different tools to each other. This is not a problem that I'm going to be able to solve on my own. But I think from our perspective, there's basically two things that can happen. So one is the sort of rote canonical answer of more and explicit tests and test cases would be useful overall. And that applies not just to sort of individual pieces of code, but kind of end-to-end testing of the results themselves. The other thing I think that's really the case, and this is almost more of a social issue, I think often bioinformatics and genomicists suffer, and there's a few that I could name, but I won't, suffer from this inability to see a distinction between the method and the code. So what they have trouble doing is they develop a method, they publish code that embodies that method, and then they will get upset if you re-implement their method in your code. If I take your C code and I rewrite it as a Spark or Scala or Java or whatever program, they get upset this is a fundamental blocker to exactly what you're describing. So if they can't even see the distinction between method and code, they're going to have trouble understanding when it's the same method with functionally equivalent results implemented in a different environment. For them, equality of the method is equivalent to equality of the code. So the only way that I know how to do this is to try to get out in front of the problem and define some of those tests myself. So re-implement a method, say from cancer genomics in Spark, show with my own methods and with my own tests that it's equivalent in some way that I define or that we define, and then go from there. Basically challenge them to tell me why it's not as good, and then we can move on. Oh.
Yeah. So I happen to have a partial answer, at least, to that question.
Okay.
The answer is develop it longer. Keep developing the software project a little bit longer than the other guy who has a method that is going to switch for the most performant one. Case in point, Snap. It's a reference liner done by UCSF and MSR based on ADAM. But
So actually, it's not based on ADAM. Snap is relatively different from ADAM, but that's a small distinction.
It's using Spark at least.
Okay.
And its accuracy is currently put into question because there's just a better research paper that has yet another method that bumps the accuracy by a few percent. Therefore, Snap is quasi abandoned. The answer to that is keep developing Snap and improving it.
Yeah.
I had another question. What is going to happen with avocado? Mount Sinai, right here in New York, has another variant called a project called Guacamole, inspired by avocado. And I see your liaison on that slide, and it seems that AMPLab is his former affiliation.
I think Uri has a different affiliation. That's probably a specific question we should take offline.
Former, yeah.
Yeah, we should take that offline.
Hey.
Hey, Josh.
So solving the yet another workflow problem isn't just building, well, I won't say building a better workflow, but separating the platform from the workflow. It's also distributing the platform across organizations, right? And that seems like a really, really hard problem. I'm wondering if you're looking at any successful examples of that in other domains.
Of splitting the
Well, you're not right. Distributing the platform over organizations in a way, right? Everyone has to agree to use the same platform. Everyone has to agree to use the same data formats. It's not just about shipping the files. It's also about shipping the code in a way, right?
It's also about agreeing on what the previous steps were, so you can rely on someone else who's done some computation for you. And I don't even think people have the ability to do that. So one of the things that I've worked in and participated in is a multinational nonprofit organization called the Global Alliance for Genomics and Health, called the GA4GH. It's an organization in which a number of pharmas and research organizations and universities and different companies around the world are members. And it's trying to bring people together to try to define what these common steps of analysis and computation are, what the common formats and schemas are, as a baseline for the possible sharing that might occur in the future. I think they get a lot wrong. I think they miss a lot in terms of sort of the forest for the trees. But I think that's the kind of place where that particular kind of work is going to have to happen. So the agreement is not the kind of thing that I'm going to be able to manufacture with code. And so it has to happen in a kind of collaborative organization that a lot of people are bought into. Is there one more question maybe? Yeah. Hey.
Great talk. I love the concept, and I think I'm not going to be the only bioinformatician that, the light bulb goes on when they see this and really get their heads around it. But this will really take off when you start hitting that critical mass, when you start seeing the actual people that don't have the software engineering training start to pick this up. And really, Spark has this, has Python support built in, and that's the lingua franca of bioinformatics scientists, besides R and Perl and a few others.
Yeah.
So
it's a number of lingua francas, right?
Yeah, that's true, but not Scala. So So when do you see that Python? I know Adam is mostly Scala. When do you see the Python implementation happening?
That's a good question. I've mostly focused on the Scala simply because it's the language in which we can, again, for things like the region join or some of the primitives that sort of go into like variant calling pipelines. It's the language in which it's easiest for me to think about the kinds of computations we have to do. I don't think we've decided on what that like higher level interface is, and I think we're going to have to reach a point where we have kind of a higher level. These are the operations that Atom supports on top of Spark in some consistent way before we can go and talk about, well, how are we going to make sure that they're supported in Python and R and all the other kinds of adapters? That's a great question. Partly it's also driven by my ignorance of the Python and Spark bindings. But maybe we can, if I learn a little bit more, I can provide a better answer in the future. Is there anything else, or can we go get a beer? I'm really thirsty.
Okay.
Thanks, guys.